crossing over is something which can occur during meiosis, and what it does is it gives rise to a lot of variation in the different gametes, so the different sex cells that a person can possess. So this is why we see the offspring, or one reason why we see the offspring of something like animals or humans being quite varied and not clones, is because of something like crossing over, which varies the different genes which different sex cells will carry that may eventually go on to form an organism. So what we can see here is, if we've got, during prophase, our sister chromatids, let me try that again. Our sister chromatids, they kind of lie next to one another, like so. So I'll draw in blue and red to try and make this a little bit more clear. So imagine this is during prophase. So during prophase, what we'll see is our sister chromatids preparing for meiosis will be lying next to each other like so. Now what can actually happen is, during this stage, something called endonucleases can come into play, and what they can do is they can actually snip and cut these chromatids, which are able to kind of wrap around one another because of their close proximity to each other. So I'm trying to draw this the best that I can. So like so, what we can see is, and I'll highlight the kind of the bit that I'm looking at here, is this little section where two of the segments of the sister chromatids actually started to wind around or wrap around one another. So this happens during prophase 1 in meiosis, where the sister chromatids are lying close together in their pairs, and we get this wrapping around of the ends of the chromatids. So, like I said before, we see endonucleases. If they see a similar combination or similar sequence of DNA, they can actually snip, and what will happen is essentially we will get the swapping over of the DNA. So if we see the swapping over of the DNA, we'll get something that looks a little bit like this, where a segment of the blue chromosome has now attached to the red chromosome, and vice versa. So this has now given sister chromatids great variation compared to what they previously would have looked like, and the genetic information they would have contained. So we've now got a new kind of result, which is going to occur from division, as if we didn't have this crossing over of sister chromatids occurring. So now what we'll see when this cell divides into two through the first division of meiosis, we'll now have a combination of sister chromatids, which looks a little bit like this. So what we would have seen is, in this cell we would have seen all red, and this cell we would have seen all blue, but because we've got crossing over occurring here, it's varied our result, and is going to mean that there's different genetic material in those sister chromatids than we might have seen otherwise. Now, remembering that, I'm just drawing kind of our sequence of events here. Remembering now that we have a second division in meiosis, what will occur is our eventual sex cells will have different products when they divide, once again because of this crossing over. So our final chromosomes, which will have the final arrangement, will look something like this. So what we see is some different combinations. We've got some touched or unchanged, so all red, no crossing over chromosomes, which we call parental chromosomes. And that's also the same here. Whereas we've got some crossing over that has occurred, so we term those as being recombinant DNA or recombinant chromosomes. So what this crossing over has done it has caused a different sequence or a different varied makeup of DNA in potential sex cells. So these are our sex cells that we get produced, the haploid sex cells at the end. So depending on the combination of sex cells that we get fertilizing one another to create a zygote and then eventually progeny, this is going to be reflected in the different genetic makeup of the individual and give rise to a different varied organism than we might have seen otherwise.